Dear friends, we will briefly present the key facts and essential information that describe and explain Alzheimer's disease. It's true that this disease is widely discussed and that it inspires fear. This is understandable when you consider that it affects 1 in 10 people over the age of 70 and 1 in 3 over the age of 85. Its effects are devastating, as it erases all brain functions, particularly all cognitive functions and the entire intellect of the affected person, within just a few years. We now understand the Alzheimer's process that gradually destroys the brain, namely the massive degeneration of nerve cell populations. This phenomenon is called neurodegeneration. Here, we will explain how, when, and why this phenomenon occurs. This understanding allows us to anticipate a treatment in the near future. This first chapter describes how the degenerative process begins and develops in Alzheimer's disease. Here we represent the human brain and its different components. This diagram clearly shows the left and right hemispheres of the brain, separated by a fissure. We can distinguish an outer part and an inner part, which are represented in the following diagrams. Some anatomical basics before describing this neurodegenerative disease. The human brain. Infinitely complex. But its functioning is based on simple and logical principles. This diagram shows the different regions of the cerebral cortex and their corresponding functions. This is a simplified diagram, because, as you can imagine, it is the constant interaction between each region that generates the overall activity of the brain. In fact, brain imaging shows that for any specific task performed, the entire brain becomes active, with variations in activity levels between the different regions. A more detailed analysis of each brain region allowed for the distinction of 50 neocortical areas, characterized, numbered, and mapped by Brodmann. These Brodmann areas are essentially a low-resolution Google map of the human brain. Here are represented the Brodmann areas of the external and internal surfaces of both hemispheres. The frontal cortex, the temporal cortex, the parietal cortex, and the occipital cortex, as well as the different Brodmann areas corresponding to them, can be easily distinguished. These Brodmann areas, each having a relatively specific function, are characterized by a particular neuronal organization and a specific number of cell layers. The nerve cells that compose them have a distinctive shape and arrangement. The connections between neurons are organized into neuronal networks that underpin their functional role. Let's talk about neural networks. 1. It was around the year 1900 that two researchers highlighted the cellular structure of nerve tissue. These researchers were Santiago Ramon y Cajal and Camillo Golgi. To achieve this, 3 elements were necessary: a microscope, a specific staining technique for nerve tissue sections, namely silver staining, and the third element was their scientific curiosity and inventive genius. These discoveries were rewarded with the Nobel Prize in 1906. Their sketches clearly show the diversity of shapes of nerve cells, both in their cell bodies and in their long extensions, the dendrites and axons, which form the neuronal networks that transmit information. Let's talk about neural networks. 2. Understanding Alzheimer's disease, and many other neurodegenerative diseases, relies on knowing about neural networks, how they function, and how they are maintained throughout life. How are they formed? 2 principles. 1. Genetic programming for their production and targeting to different regions of the brain. 2. Their activity during brain development. Only functional neurons, those that are correctly connected to their target, will survive. They will continue their mission and organization and fulfill their role in the thoughtful transmission of information within neural networks throughout a lifetime. Those that are poorly integrated into the developing network will degenerate. And it is at this level that this precise mechanism of neuronal connectivity development largely explains what Alzheimer's disease is. A poorly connected neuron no longer receives the trophic factors essential for its survival from other neurons in the chain, upstream or downstream. As a result, it degenerates. This simple observation strongly suggests that Alzheimer's disease results from the loss of these neuronal survival factors, causing the chain reaction collapse of certain neural networks. Alzheimer's disease is characterized by neuronal degeneration with a distinct morphological and histological signature, as well as a molecular and biochemical one. 1. The morphological signature was described by Alois Alzheimer in 1904. This involves neurofibrillary degeneration. Alois Alzheimer's diagrams clearly show the accumulation of fibrils, stained with silver salts, in the cortical neurons of the gray matter of demented patients. Patients who would later be diagnosed with Alzheimer's disease. In the later stages of the disease, all neocortical regions are affected by this neurofibrillary degeneration. To be complete, we mention the other characteristic brain lesion of Alzheimer's disease, which consists of scattered deposits of amyloid substance or amyloid plaques. These deposits are observed in large quantities in the gray matter of the neocortex, and sometimes in the cerebral blood capillaries. The amyloid substance results from the aggregation of a protein fragment, a peptide called A-beta. 2. The other hallmark of Alzheimer's disease is molecular and biochemical. These pathological neurofibrils result from the aggregation of proteins. These are tau proteins, which were identified by Bryan in 1987. Here are some technical details for those interested. Tau proteins are involved in the transport of cellular materials within neurons. The aggregated tau proteins that form the pathological neurofibrils can be detected using the Western blot technique. And with this technique, the result is conclusive. Lane A. Significant detection of pathological tau proteins in Alzheimer's tissue. Lane B. No detection in healthy brain tissue. The detection of aggregated tau proteins allows for the study and quantification of neurofibrillary degeneration in all its aspects. It has been quantified in human brains at different ages and affected by various neurodegenerative and other diseases. It has also been investigated in other vertebrates. Based on these studies, the following observations have been made. Firstly, Alzheimer's disease is specific to the human species. No trace of aggregated tau proteins has been found in great apes. Secondly, Alzheimer's disease is a specific type of dementia, different from other neurodegenerative brain pathologies, including Parkinsonian syndromes, frontotemporal dementias, and others. Finally, Alzheimer's lesions with aggregated tau proteins are not detected in other organs, which explains the difficulty in making a biological diagnosis. The ultimate conclusion for this first part is that Alzheimer's disease is characterized by two different brain lesions in the neocortical gray matter. These two lesions coexist, but there is no strict spatiotemporal relationship between them. Genetics indicates that the cause of the disease is linked to amyloid substance, aggregates of the A-beta peptide, since mutations in the amyloid precursor protein are responsible for familial forms of Alzheimer's disease, as demonstrated by Hardy in 1991. It was logical to follow this lead in the search for a treatment. Unfortunately, this has resulted in 35 years of repeated failures. However, by following the trail of the tau protein, as we will attempt to demonstrate, it is possible to find a comprehensive and logical explanation that accounts for both of these lesions. This tau protein pathway leads directly to a therapeutic strategy. Moreover, it has recently proven successful in a clinical trial for a rare disease related to Alzheimer's disease, another tauopathy called progressive supranuclear palsy. Since tau is a reliable and accurate marker of neurofibrillary degeneration, it became possible to quantify the Alzheimer's degenerative process in a comprehensive and complete manner. How to proceed? It was, of course, necessary to have autopsy samples for the measurements. And for each brain, to quantify the tau pathology in each Brodmann area. And to have brains from patients of different ages, as well as patients at different stages of Alzheimer's disease, and also those with other neurodegenerative diseases. Therefore, a brain bank was needed, which we were able to establish in our INSERM unit in Lille. Of course, it was necessary to cooperate with various hospitals, especially in Lille, but also nationally and internationally. Because, importantly, for each brain, we needed to have the corresponding clinical records, which we successfully obtained. This graph represents a study conducted on several hundred patients. Each row shows the number of the Brodmann area analyzed. This allows you to see the presence of tau pathology and even its intensity. For example, in the third row, for patient S7, you can observe that the tau pathology is beginning to spread into areas 20, 21, and 22 of the temporal cortex. It is therefore possible to determine the progression of tau pathology during Alzheimer's disease. And this is for a specific reason. This progression is unique and invariable. Our study makes it possible to determine the progression of neurofibrillary degeneration at different stages of Alzheimer's disease. This is possible because, as we will see, this progression follows a very specific, constant, and invariable path. We have identified 10 stages of progression, which correlate perfectly with the clinical decline of the patients. Here, in this representation, we are at stage 0. There is no detection of pathological tau proteins, regardless of the region studied. This is what we observe in control subjects under 75 years of age. All individuals over 75 years of age have tau pathology in this region. This represents an unavoidable vulnerability of the human brain revealed by tau pathology. This region is part of the limbic cortex, the cortex associated with memory and emotions. No detectable impact on brain function. The second region to be affected is the entorhinal cortex, Brodmann area 28. This region is connected to the first one through its neural network. Stage 2 appears to be clinically silent. The third connected region is the hippocampus, Brodmann area 34. The hippocampal region is vulnerable to tau pathology during aging. This tau pathology of brain aging can occur with or without amyloid deposits, the other hallmark lesion of Alzheimer's disease. The hippocampal region is particularly involved in short-term memory. Subtle cognitive impairments are likely at this stage 3. Cognitive tests can objectively assess these cognitive impairments, which may fall into the category of mild cognitive impairment. After the hippocampal region, the process of neurofibrillary degeneration progresses into the anterior temporal cortex. This Brodmann area 38 is also a limbic region, like the hippocampus. At this stage, discrete deposits of amyloid substance are generally found, indicating that the Alzheimer's process has begun. The first language difficulties may appear, in addition to memory problems. The patient enters a stage known as mild cognitive impairment. Numerous cognitive tests can objectively assess and quantify this stage, such as the Mini-Mental State Examination, the Montreal Cognitive Assessment, the Clock Drawing Test, or the Du Bois 5-word test. Next, the inferior temporal cortex is affected by the neurofibrillary degeneration process. Amyloid deposits, aggregation of the eta-42 peptide, are present. Cognitive impairments in memory and language intensify, marking this progression of Alzheimer's disease. Stage 6. The middle temporal cortex is then affected by neurofibrillary degeneration. This represents a progression from stage 5, both in terms of tau and amyloid pathology and clinical symptoms, primarily memory and language impairments. Stage 7. Neurofibrillary degeneration will simultaneously affect all polymodal associative cortical regions, namely the anterior frontal cortex, the superior temporal cortex, and the inferior parietal cortex. Amyloid deposits are significant. At this stage, cognitive impairments progress to the 4 A's. Amnesia, memory loss. Aphasia, language impairment. Apraxia, difficulty with motor skills. Agnosia, difficulty recognizing objects. Stage 8. Next, the less associative cortical regions, known as unimodal areas, are affected by the escalating degenerative process, such as Broca's area, namely Brodmann area 44, the motor speech area. It is worth noting that the progression of the tau pathology is paralleled by an intensification of the degenerative processes in the initially affected regions, such as the hippocampal region. The worsening of clinical symptoms goes hand in hand with this cerebral invasion by the degenerative process. Stage 9. Tau degeneration will then invade the primary cortical regions, including the visual and/or motor areas. Amyloid deposits are very significant. This is a severe stage of Alzheimer's disease. Intellectual, motor, and sensory functions are severely affected. Stage 10. This is the final stage of tau degeneration, as all cortical regions are affected, as well as numerous subcortical nuclei. This represents the terminal stage of Alzheimer's disease. No brain region is spared. Amyloid deposits are also very extensive. All cognitive functions are destroyed. Conclusion. This progression of tau pathology, following a relentless path throughout the entire neocortex, in parallel with cognitive decline, is both evident, understandable, and undeniable. The correlation with the assessment of cognitive abilities is relative. This is understandable. Indeed, when quantifying a lesion process, it is impossible to estimate the number of still functional neurons capable of compensating. But the main information, in our opinion, is the following. The degenerative process as we see it corresponds to a chain reaction of destruction of a particular neuronal network. We mentioned at the beginning of this video that neurons only survive if they are connected to those upstream and downstream for the exchange of neuronal survival factors. If, at the beginning of the neuronal network chain, a group of vulnerable neurons, for example, the hippocampus, begins to degenerate, it will gradually drag the entire neuronal network down with it. This is all the more true given that other neurodegenerative diseases exist, and they all correspond to the destruction of specific neuronal networks, which characterize each type of disease. All of this leads us to a therapeutic hope, which is the subject of our last slide. Final conclusion. In summary, Everything seems to indicate that the cause of Alzheimer's disease results from a deficiency in neuronal survival factors in a neuronal population vulnerable during aging. These survival factors could be metabolic products of the APP protein involved in Alzheimer's disease. This is our hypothesis following the comparative analysis of human brain tissue. A screening of a chemical library allowed us to find a molecule that stimulates the potential survival factors derived from the APP protein. This molecule is called AZP2006. Great news! This molecule reduces tauopathy in animal models. Furthermore, AZP2006 yielded excellent results in a therapeutic trial on a human tauopathy, progressive supranuclear palsy. The circle seems to be complete. A molecule targeting APP protein that acts on tau pathology. This reconciles the two lesions of Alzheimer's disease, the amyloid beta hypothesis and the tau hypothesis. This gives us hope that the therapeutic target for Alzheimer's disease has been found.